When I got the invitation from the GCC, I was looking forward to it. Wanted to show you some of Merck data, but when I realized that Fadi from Pfizer was going to be here and Chinzia from Genentech, I decided to take a different approach and talk about some of, I think it's actually more interesting to talk about this than talking about what Merck is doing. Sweet. Okay, so briefly, I'm going to, since this is an ADME drug discovery type symposium, I'm going to briefly touch on it. Then I would talk about methionine aminopeptidase, which is one of the things that I worked on in grad school. And then I'll tell you some of the characterization that we did at the Center for PKPD. All right, so you've seen this in so many different forms, and you're very familiar with it and how drugs go through the different stages of development until one of it eventually makes its way into people and become a drug on the market. But I want you to remember that before you get to that point where you get one of those molecules to become a drug on the market, you have to have a fine balance between all these different, I don't know what to call it, but you have to have a very fine balance between all of this. You know, you have to think about the ADME properties, toxicity, safety, PK, bioavailability, so many things until you select that one candidate that eventually would make its way and hopefully become a drug. So earlier I wanted to do a quick survey to see what people's thoughts are on this. So quick question, why do drugs fail? Now, Wang from Novartis and Urban from Yale in 2004 had published his paper, more or less a retrospective study of failed molecules at the recent time, thinking about why do these molecules fail. If you look at the plots that he had at the time, he had put it that 39% of the molecules that had failed was because of the poor ADME PK/PD properties. Lack of efficacy was also up there. The rest of it had influences, but most of it was ADME and lack of efficacy. Now, fast forward to 2019, I would say that the common industry perspective today is that drugs are no more failing because of a lack of understanding of the ADME properties. We've done a very good job of understanding ADME. Drugs are currently failing because we have a very poor understanding of the underlying disease condition, and we also do not have proof of concept in humans before we take these molecules into people. So I put in here a typical research operating plan to show you the bunch of different ADME profiling assays that we often run before you put a drug in people. You would see that for you to run all these different cycles, this is not for you to read, it's just to show you that it gets really crazy. For you to run this, you would estimate that you would spend at least $300,000 for you to run one molecule through this ROP. Now, if you take what I just showed earlier and just summarize it into three buckets, you know, in lead ID and optimization, we run a bunch of very simple screening assays, rodent, non-rodent, PK. We would want to see CYP liabilities, think about absorption. You want to see if it's a PGP substrate. You think about plasma protein binding, and also see if it's partitioning between blood and plasma. Now, when you move towards single candidate selection, then you have more definitive studies where you spend a lot more money. You know, you have to do definitive rodent and non-rodent PK. You have to characterize the plasma protein binding in different species. You do mass balance studies. It could be hot sometimes. Sometimes you could get away with doing it cold. You think about recombinant CYP phenotyping. You do UGT phenotyping as well. It gets a little bit more. You spend a lot more money at this phase. Now, as you select your preclinical candidate, then you do some purpose-driven ADME studies, hot or cold blood partitioning, hot or cold BDC, you do your MetID profiling, then you spend some money in model-informed drug delivery, and then maybe SimCyP, PBPK. Now I'm going to switch gear a little bit, having talked about all that, to talk about CLBQ14, a very interesting molecule that is targeting methionine aminopeptidase for infectious diseases. So why is this important? Now, it is well reported that antibiotic resistance is a thing, right? in recent times, if you think about tuberculosis specifically, the FDA have approved only one molecule in the last 40 plus years. So I don't know whose kid this is, but I thought this was very interesting. So is TB really still around? Absolutely. So as of 2014, the CDC put it that 1/3 of the world population is affected by tuberculosis. That's 1 in every 3 person. So it's still a thing. And if you fast forward and think about patients living with HIV, people living with HIV, sorry about that, you would see that extended drug-resistant tuberculosis is one of the leading killers in this population. And then, of course, the rise in TB resistance is well reported, and its impact is very obvious. Now, we're still treating TB the same way we did 40 years ago, right? And that's the right approach. We need to start thinking about new drug development, and thankfully, Dr. Olaleye had spent the majority of our time at Johns Hopkins doing that. So again, to reemphasize the timeline for TB drug discovery, you could see that between 1963 and 1994, about 30 years gap, there was nothing that was made for this population. And there was also that big gap between 1998 and 2012 where nothing was done until bedaquiline was brought onto the market. Now let's talk about methionine aminopeptidase a little bit. I feel like everybody in this room understands the central dogma. I'm not going to tell you about it again. But if you think about what happens after that, when you have the first step, you often have a formulated methionine polypeptide. Now, PDF comes in and chops off the formyl group, and then you have a methionine polypeptide. Now, MetAP comes in, chops off the methionine, and then you have proteins, polypeptides that would then undergo post-translational modification, stabilization, and all of that. You have fully functional proteins and polypeptides. Now, if you look at this already, you could say, oh yeah, there are 2 targets that are possible, right? If you block that first target, PDF, then you could probably get a drug. Or if you think about blocking methionine aminopeptidase, you might also get a drug. Now, PDF inhibitors had failed in the clinic, and methionine aminopeptidase became a very interesting target for a lot of people. How important is MetAP? So when you think about it, it's been published as far back as 1989 that deleting this target is very lethal in microorganisms. Now, all these different people have done all sorts of work just validating this target for these different infectious agents. Now, it's also been looked at for other diseases, cancer, it's been looked at for rheumatoid arthritis, it's been looked at for malaria, fungal infections. People are really spending a lot of time looking at this target. Now, Dr. Olaleye at the time at Johns Hopkins had screened about 175,000 compounds against MetAP in TB, targeting 1C. And she had set limits of 30 to 40% inhibition as potential hits. Now, of so many that she probably had found to be very interesting, this were the most interesting of all of them. So CLBQ14, when you put this into co-culture and just look at CLBQ14 in TB, you would see she had found very interesting MIC and very low inhibitory concentrations compared to cleoquinol. Now, cleoquinol is an old drug that was previously used for travel diarrhea, but it had to be taken off the market because of CNS toxicity. But currently, I think it's being used topically for infectious diseases, but nothing orally. So this, if you look at both structures, they are very similar, right? They only differ in the position 7, whether it's a bromine instead of an iodine in position 7. Now, Dr. Shea's lab. That's when the molecule came to Dr. Shea's lab to see what can we do. Now, when a molecule comes, first thing we do is to be able to develop tools to quantitate the molecule, because if you put it in animals, put it in whatever system you want to put it in, you have to be able to quantitate, and you're as good as your numbers are. Now, we develop a UV method, develop an LCMS method to be able to quantitate this, do the full validation according to the FDA guidance, and put out a paper on this. Now, another thing we did was to find out what the thermodynamic stability was. So we put it in a bunch of different solvents, and you could see that the solubility in water was extremely low. And then we thought of ways to put this in animals, and co-solvent is one of the strong pillars in the group, and we developed two co-solvent formulations to put this in animals. Now, one of the concerns we had at the time was aqueous stability for this compound, so we exposed it to different pH range and wanted to know how stable is this compound over time. Now, if you look at this, it doesn't make any sense, but if you take that apart and plot the K constant against the pH, you would see that at lower pH, your drug is pretty stable, but as the pH increases, it falls apart. So we went ahead and put that in rats and wanted to know what the disposition was. We put it in IV, couple doses, just wanted to see if there was a dose linear relationship, and we were able to see that it was pretty linear. I mean, I guess we could expand these doses and look at higher doses, but I mean, at MIC concentrations that were very interesting, this dose was good enough. Now, if you take that and put it in rats and try to see what the bioavailability was, we saw that subcutaneously or in oral routes, you get pretty good exposure. Now, if you summarize the PK and see if the numbers really make sense, the Cmax was pretty good. Tmax in oral and subcutaneous were less than an hour. And when we looked at the clearance, you had pretty decent clearance. The T-half was about 8 hours. Now, bioavailability-wise, PO, we found about 40% bioavailable, and subcutaneously, we saw very high numbers. Now, I strongly believe that the subcutaneous bioavailability was significantly increased because we used transcutol, which everybody knows would open up the skin for subcutaneous absorption. Now, we characterized a couple other numbers that we feel were important for us to use to explain the PK. Looked at plasma protein binding, blood partitioning, and looked at the log P. Now, when you take animals down at 2 hours and collect the tissue, we wanted to see what the exposure was at 2 hours. Now, if you're targeting infectious, the target is MetAP for infectious diseases, you want to be able to touch all these different organs and see if you're getting enough exposure. And at least at 2 hours, so we're able to see pretty good exposure. We had concerns about the kidney because it's highly accumulated in the kidneys, but I believe with mass spec imaging, we should be able to tell and see if it's in the cortex or the medulla, and that will inform on if it's going to be renally toxic or not. So if you calculate the tissue plasma ratio, you could see it's highly accumulated in the kidneys. Unfortunately, we saw that less than 1% of the drug was excreted on change in urine, so what we're seeing is not because it's in urine. So then we put this into a in silico model, trying to see what the soft spots were for metabolism, and you could see that position 3. Just by looking at the molecule, you could see position 3 and 6 are very likely soft spot for these molecules to be metabolized. Then we put this in liver microsome and wanted to see from the different species, can we get any interesting microsomal metabolism? And we saw that in mouse, as Stan had talked about earlier, that mouse just chews up this compound. So if you take that down and just plot it after 60 minutes in incubation to see how much was remaining, you can see mouse chews it up. Some of it was left in rat, and some of it was left in human microsome as well. Now, from this, you could definitely calculate what the clint is, and predict what the hepatic clearance is going to be. Now, if you take those numbers and put them in your well-state model, correcting it for plasma protein binding and also for how much of it is exposed to blood plasma partitioning, you could definitely predict what the human clearance is going to be. Now, if you compare your clearance numbers to your hepatic blood flow, which is what we did here, we could see that the clearance is well less than hepatic blood flow, meaning that most of the drug is not being chewed up completely by the hepatocytes. Now, another thing we were interested in seeing potential DDIs and seeing if you put this in different recombinant human SIPs, which of them was chewing this compound up. And you could see that if you take it down to at 60 minutes and plot and see which of this, you could see that 1A2 and 2D6 are the potential SIPs that would metabolize this compound. Luckily for us, we don't have any CYP3A4 metabolism. Well, at least we think so from the in vitro study that we've done. So at least there's no likelihood of a DDI from C3A4. So in summary, we have some key questions that you still have to answer. And I guess just switching from being a grad student to industry, I know that this compound is a very long way away from getting into people. You still have to talk about in vivo efficacy model. Still have to think if, fine, this compound is not a substrate of 3A4, but is it inhibiting it or is it inducting CYP3A4? Of course, you have to phenotype for UGT and also think about oral PK from potential salt that might have increased solubility. Definitely, you need to do PK in higher animals, and you still have to think about tissue binding and know what impact that might have on the toxicity. Most of the people that worked on it have all moved on to work for different companies, including myself, but I still want to acknowledge all of them for their contributions. Thank you very much.
The question I have is that, pardon my aging eyes, okay? So, because you and I have a 60 years of difference. I didn't see when you had a slide on TBs and you don't have to flip there, you just have to answer me. And then with the TB there, I saw a lot of, you had the companies and whatnot in there who were working on TB compounds and whatnot, right? Yeah. But I didn't see, did you have it on a nonprofit organization?
No, I didn't. That was everything.
Good.
I pulled that from a paper.
Very good. Yeah. This is a blind spot because I'm talking to the whole audience too, because drug development is not just companies or academic settings. At least there's some nonprofits are also into drug development. Pardon me, I really am speaking as a private citizen, and I'm not for this company, but Bill and Melinda Gates Foundation, they have it. I know that they're working on this area, and also they're working on like malaria and all that, so I just want to let the audience know. I like to think out of the box. You know who I am.
Thank you.
We got you, Yoli.